Bacterial spore or endospore. It's a highly resistant resting phase, the resting phase of bacteria during abnormal environment. That means this is a resting phase of bacteria. That means when there is a lack of nutrient or high temperature, if the bacteria is not getting proper nutrition for their growth, what will happen? This bacterial normal vegetative cell will be converted into endospore formation. The bacteria will be converted into endospore. In this endospore formation, due to this endospore formation, the bacterial DNA can be preserved and other cellular organelles can be preserved. And this will, in this condition, the bacteria will be resistant to heat, chemical, radiation, and desiccation and all. That means during this endospore stage of the bacteria can be live longer for years and years. So this endospore will resist heat and chemical or high cold condition. And endospore are called as spores inside the bacteria. That means endo and spore. That means spore inside the bacteria, we will call it as endospore. And one bacteria can seed only one spore. The examples are Bacillus species. This is the Bacillus species, Bacillus anthrax, Clostridium, and Sporosarcina species will have these endospores. Structure of endospore. Here you can see this is the structure of endospore. Exosporium, spore cord, next cortex, core wall or spore wall, and later inside you can see this is the core. And the exosporium is made up of lipopolysaccharide, and all the organisms, all the small spore-bearing organism doesn't have this exosporium, and some spore-bearing organism will possess this exosporium. And just inner to exosporium, you can see this is a spore cord. The spore cord is made up of keratin-like proteins, and it is impermeable. That means this spore cord will prevent the spore from harmful chemicals. And inner to the spore cord, you can see this is the cortex. This is the thickest layer of this endospore, and it is made up of polysaccharides. This cortex is made up of polysaccharide. And inner to the cortex, you can see this is the core wall. Core wall is also called as spore wall, and it's made up of peptidoglycan layer. Later, this peptidoglycan layer will become cell wall when it will become to vegetative cell. And the innermost layer, you can see this is the core, and it's also called as spore protoplast. And this contains nucleus and also some enzymes such as dipicoic acid. Shape and position of spore, you can see this is the first one is spherical central spore. That means the spherical shaped spore will be seen in the center of the bacterial cell, and it is non-bulging type. And this is oval. Oval central means the spore shape is oval, and it is located in the center of the bacterial cell. And it is oval subterminal. That means the structure of the spore is oval, and it is present in the subterminal area. And we can say that these three are non-bulging. That means you can see the spore is seen inside the cell, and it is not projected outwards the cells. That is why it is called as non-bulging spores. And next about the bulging spores, we can see that this is a oval subterminal. Same thing only, oval subterminal, but it is bulging. It is oval terminal. The spore will be seen in the terminal portion, but it is bulging. And the spherical terminal. Spherical terminal means the shape of the spore is spherical, and it is present in the terminal portion of the bacterial cell, and also it is bulging type spore. The last one is free spores. Free spore means the spore is not present inside the bacterial cell, it is seen outside the bacterial cell. Sporulation. Sporulation is a process when there is an unfavorable condition for the bacteria, mainly the spore bearing bacteria, that will convert the vegetative cell into spore formation. So this spore formation consists of different steps. In step 1, we can see the axial filament of bacterial DNA is formed in the step 1. When it goes to the step 2, we can see here in the cytoplasmic membrane or plasma membrane, a septa will be seen. That means the septa formation will be occur. From this region, what will happen? The cytoplasm divides and a 4 spore is seen here. Now we have one 4 spore and this one is the mother spore. And the another step, what will happen? This mother spore will engulf this 4 spore. In the third step, you can see here, there is a 2-layer of plasma membrane can be seen here. And in the next step, what will happen? The quarters formation can be seen here due to the accumulation of calcium and dipercholic acid can also be observed here. And in the next step, you can see the protein coat is formed in this 4 spore. And the next step, what will happen? The step 6, we can see the maturation of spore can be occurred in the spore 6. And in the step 7, the liberation of spore from this vegetative cells will be occurred. And these are the cycle of sporulation. First, starting with the step 1, that formation of axial filament of bacterial DNA is formed. Next, the septa will be formed. The septa will be divided. It will get 2 spore, that is 4 spore and mother spore. Later, this mother spore, cytoplasm will be engulfed by this 4 spore. In the third step, we can see the 2-layered plasma membrane, and the step 4, the cortex is formed between this plasma membrane, and also the calcium and dipercholic acid can be observed here. In the 5th step, the protein coat is formed, and in the 6th step, the maturation of spore will be occur. At last, the liberation of spore will be seen in this last step. Germination of spore. The germination of spore occur when bacteria will get a favorable condition for their growth and metabolism. In artificially, we can do this germination process by heating this spore. While heating this spore, what will happen? This core of this spores will be hydrolyzed. That means the core will be damaged. When the core will be damaged, the next step, the initiation process will be take place. So what will happen? It will increase the metabolic activity. And again, when we are giving water to these spores, it will release calcium and dipercholic acid. And after that, the outgrowth will be take place. That means during this outgrowth, cell wall synthesis, protein synthesis, and enzyme activity will be at high level. At last, the spore will be converted into vegetative cell. That means this germination process of spore is a reversal process of sporulation. The resting phase of the spore will be converted into the vegetative phase of this bacteria.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.